Hello, and welcome back to a fresh day of scientific ethics, where we're going to start to apply some of this information about our DNA molecule. And in our first way of applying our information about the DNA molecule, we're going to take a look at DNA fingerprinting, because this is an application that's used pretty extensively around the world, and it's something that you can actually use the concepts in a take-home set. So you can, buy a kit for about $90 and have them go through and take a look at your DNA sequences, and they'll give you an idea kind of who you're related to and kind of what's your ethnic background and a lot of things like that. And so we're going to take a look at kind of how this actually works. And so DNA fingerprinting. Now, as a review, remember that what we're talking about is that DNA molecule is a double helix. And so the important things that we're worried about, the A's, the T's, the G's, and the C's, are going to be those nucleotides towards the inside, kind of the rungs on our ladder that we're focusing in on. And again, we've got the backbone, the kind of sides of our ladder kind of over here. The A's, the T's, the G's, and the C's are on the inside. The A's bind to T's, the G's bind to C's. But this flattened molecule is actually going to be twisted around into a double helix in the actual molecule. And as we talked about, the information is going to be coded into the DNA molecule, into one of these strands in the sequence of the A's, the T's, the G's, and the C's. So the sequence of the A's, the C's, the G, I'm sorry, that's a T, and the G's, it's going to hold that message. So think back to the, I can't remember what it was, the big cat ate the red rat, red dog, something like that. Should remember it. But basically, it's going to be three-letter words, or the equivalent of that in the DNA molecule. And so what we want to do is try to understand what's going to be going on with this. Okay, so the first thing we're going to do is we're going to go through and say, okay, we're going to have a mechanism that we're going to cut up our DNA molecule. Now, this isn't something that you normally want to do. And so what we're going to be looking at, again, the terms aren't as important as the concept. So we're going to be looking at what are referred to as restriction enzymes. These are a natural defense mechanism for bacteria. Think about bacteria, they're going to go around, they're going to expose to a lot of things. And so potentially, they're going to be very good at picking up foreign pieces of DNA. And those foreign pieces of DNA could potentially cause an infection or screw up the cell in some way. And so their defense mechanism is that they're going to have these enzymes that are going to go through and help break down that DNA molecule. And so what's going to happen is they're going to go through and break up the DNA molecule at specific sites. And again, don't worry about the names of all of these different things here. But what we've got is basically our ladder, kind of instead of running like a ladder up like this, we're going to have the ladder kind of running along the sides. I don't know if I can do a pointer or draw on it. Maybe try a pen. So, instead of our ladder kind of being up like this, like our previous slide, what we've got here is the ladders are running kind of this way. So, I don't want to do the pointer anymore. So we got the A's, the G's, the C's, and T's. A's bind to T's, G's bind to C's. And what we have with the restriction enzymes is it's going to cut at this specific point right here and break the molecule off into two parts. Another one to break it here. This one kind of breaks it along this point through here. It's kind of neat. I can go through and do this. But basically, the important thing is that these restriction enzymes are going to go through and cut the DNA molecule at a specific site. I should probably clear this. And so we can go through and know that anytime we have this sequence, we're going to cut the DNA molecule here. So what's going to happen then is we're going to combine that technique of cutting the DNA molecule up with a process called gel electrophoresis. And so what's going to happen here is we're going to cut up the DNA molecule based on the sequences, based on those restriction enzyme sites, and then we're going to load them on the gel here. Kind of like the equivalent of scientific jello. And so there are going to be little spaces in here. So we're going to load the DNA molecules up here. We're going to run an electrical charge through this, and the DNA is going to move down in this direction. And the way I describe this in the class is that this is almost like driving on I-95 or driving on a highway in rush hour. And so big molecules up here, you know, big molecule kind of like this is going to take a long time to move down through here. So a big semi-truck, you know, taking a look at some of the semi-trucks that we see, you know, with tandems, you know, running, you know, one cab with two trailers, they're going to have a hard time getting through traffic. But you get a little sports car, you know, a little DNA molecule like this, they can kind of zip in and out of the holes as it's going to go through. And they're going to move down I-95, kind of down towards Baltimore relatively quickly, where our semi-truck is going to, you know, take a long time because it's not able to get into those little openings. So it's going to move slower down through this. So what this means then is that through the process of gel electrophoresis, we load all of the molecules up here at the top in one of these regions up here. The big ones are going to come through slowly. The little ones are going to move through very quickly. Kind of medium-sized ones, kind of like this, are going to be kind of in the middle. And this is going to allow us to separate our DNA molecules by size. Now, again, if we keep running this, you know, like you know, rush hour, even though it doesn't seem like that at times, you know, that semi-truck that's moving slowly down rush hour, moving slowly down I-95, is eventually going to get to Baltimore. It's just going to take a lot longer than the little sports car that's zipping in and out, changing lanes along the way. What this means then is that we can separate our pieces of DNA based on the size of what we've got present. There's those. Go to the next slide. So what we then see is, instead of looking at it like this, where we load the DNA molecules in, they move down with the current, we can stain with a variety of sorts. In this case, we've got this kind of fluorescent dye over here that we're looking at. This one, we're looking at another mark here, maybe a fluorescent dye that we're taking a black and white photo of, or you're using some type of reader. And what we can see is that these are the places where we load the DNA in up here. We run the electrical current through this. So this represents our scientific jello, the gel that we're talking about. And so these are going to be big molecules over here, medium-sized molecules down here, and tiny molecules here. So we're able to separate them by size. And that's interesting. A lot more interesting when we apply it. But we can look at it and understanding the pattern, we can learn a lot about the DNA of the cell or DNA of the individual that we're looking at. Now, the most common use of this is this idea that each individual is going to have a unique pattern of DNA fragments. And this is going to be referred to as a restriction fragment length polymorphism. An incredibly complex word, but take a look at it. RFLP, restriction fragment. So restriction are like the restriction enzymes. It means that we're going to be chopping up our DNA molecule into little fragments based on the restriction enzyme we use. So we're going to be chopping it up, restriction fragment, and we're going to have a length polymorphism. Again, fancy words, but take a look at it. Length just means that the length of the molecule, or the length of the fragment that we're looking at is going to be polymorphism. Poly for many, morphism for shape. Basically means that each individual is going to have a unique pattern of DNA fragments. So they're going to have fragments of varying lengths as they go through. And then we can use that to find out information about the individual. In some cases, if we know information about these different lengths of the DNA fragments that we're looking at, we can often find a nearby allele, even if it's not showing up exactly where we want. It could be identified on the portion of the DNA molecule that we're studying. So each individual is going to have a unique pattern of these DNA fragments. And so if we take a look at this, its first use was to do paternity. And so if we take a look at this, each individual is going to have a unique pattern, okay? And so this is looking at the child, the mother, possible father 1, possible father 2. And so we're going to chop up their DNA using the restriction enzymes. We're going to load it on our scientific jello, our gel electrophoresis, run electricity through it, and we're going to have big molecules at the top, little molecules down here at the bottom, again, because it's going to separate these based on size. And what we're going to see is we know that in this case, this child has two fragments that we're looking at. I'm oversimplifying this, but you get an idea from it. And they're going to have one fragment which is going to be coming from the mother, and one fragment that's going to be coming from the father. And so if we take a look at this, we know that in this case, the child has two of these fragments, one fragment from the mother, one fragment from the father. Now, if we take a look at the mother's DNA, her pattern of these restriction fragment length polymorphisms, or at least the different sizes of the molecules, different bands that we're taking a look at. She got a heavy one up here, a big one, and a kind of medium-sized one up here. This medium-sized one here kind of matches up with this medium-sized one that the child has. So in this circumstance, this is the information that the child has received from the mother. And again, that mother, the heritable trait, in this case, the heritable trait is the pattern of the DNA molecule that's been cut. So in this case, this one is coming from the mother. And then we take a look at the other one and say, okay, where's this coming from? And we take a look at it. Possible father number one doesn't match up with any of these, okay? Possible father number two has this situation where we've got a match down here. So that means possible father number one isn't a possible father. Possible father number two could be the father. So we can exclude this person and say he's not the father. We could say, based on this, this person could be the father. We probably need to do more tests. Doing it on a single thing like this isn't scientific, but if we were to do more tests, we can determine whether or not this is likely to be the father or not. And again, this is based on this pattern, based on the fact that the offspring is going to receive one characteristic, one trait, one DNA, one gene from the mother, and one gene from the father. Oops. Go in the right direction. So again, what we're doing on this is looking for shared bands between the parents, match up the mother and the child first, and then find the father's band matching up with the child's band. So again, eliminate that person and make sure that this one is a possible parent. Now, similar DNA techniques are used to trace ancestry. And so if you do an ancestry kit or 23andMe or any of the variety of them, you can see them advertised on the internet, you can see them on social media, very prominent in the holidays. You get one of these kits, you basically leave a DNA sample. So they have you put your saliva in a tube. They will then take that back, run the DNA, the same way we're running it here, run this, look at that pattern, and then run it through a great big database. And then based on that database, it's going to give you information about who you're most closely related to. Again, the person you're closely related to is going to have the most similarities to you. And you can find, cousins and second cousins and further relations like that. And you can also look at, where your family is likely to have immigrated from, if you're coming to the United States from another country, what's your heritage, things like that. So again, it continually is being improved, but using these techniques can be used to trace your ancestry. We can also use this in forensic DNA testing. And so we can see this occurring, see it on CSI movies, things like that, where instead of looking at, a paternity pattern here, we're going to be looking at the same thing. We've got the defendant's blood, we've got blood from the defendant's clothing that he was wearing when the police found him. I can't find my pointer. And then we've got the victim's blood. And so we did the same thing. We took the victim's DNA and the defendant's DNA. We chopped up the molecules, loaded on our fancy scientific ran some electricity through it, separated the bands based on size. And what we can see is this is the pattern of the defendant, and this is the pattern of the victim. And so if we take a look at, the bloody shirt that this person was wearing, we take a look at it and say, okay, does this pattern of DNA fragments kind of match up with the defendant, or does it have some similarities to the victim? So in this case, when we take a look at it, we can see, almost a one-to-one relationship between all of this blood, which is found, all these fragments of DNA that are found within the blood of the victim was found on the bloody clothing of the defendant. And so this is going to be very good evidence that if we look at enough of these bands and enough of these patterns, we can run statistics and say it's highly likely that the blood found on the defendant's clothing came from the victim. And this is often used then in a court case to show, that the defendant was likely the perpetrator that, attacked the victim in this case. We can also look at what are referred to as short tandem repeats. So if you think back to when I talked about the DNA molecule in the last class, I said that I oversimplified it a lot, that there were, some regions that code for information for how to build the proteins. There were regions of the DNA molecule that allow you to turn on and off or enhance or promote it, so turn it on and off and make lots of copies or a few copies. But there were also sections that just had these repeats that were present. And so these short tandem repeats are going to be short segments of DNA that have many, many different polymorphisms. And so individuals have, between 1 and hundreds or thousands of copies of this. And so they can go through and study these polymorphism patterns and based on statistics, determine how likely it is that two individuals would have the same pattern. And so the FDI uses 13 different locations within the chromosome molecules that they're looking at. And at these 13 different locations, there's a lot of polymorphism. And based on the statistics, two people having the same pattern for all 13 of these, the 13 that are here in yellow, are basically about 1 in a billion. And so if we're looking at, a planet that has about 9 billion, I think, people on it now at this point, the exact pattern solely by chance is going to be very, very rare. You'd have to find 9 people having the same pattern across the entire planet, unless they're identical twins. Keep in mind, identical twins have the same genetic information coming from parent to offspring. And so because of this, the FBI system running through this, referred to as CODIS, basically says that if they come up with a pattern, it is very, very likely that they can match it up with an individual and identify that individual having this pattern. Now, simply doing that isn't enough. They're still going to need to do some additional evidence to, show that they're, the things that lawyers do and, police investigators do. But this can help identify an individual if they're not sure who the victim of a crime or who, could be doing the crime. They could take a look at the DNA and try to identify people. Now, then the question then becomes, if we use this CODIS, this combined DNA index system, which is what CODIS stands for, it then becomes a criminal justice DNA database. So it's almost as if you're putting kind of this barcode onto everybody that we can then use to identify them. Because if we get a sample of your DNA, we can run it with the same sequence of chopping it up with the restriction enzymes, running it on the gel electrophoresis. We can do this multiple times, and it's going to show up and give you that same pattern every time. And so CODIS then is this national database that is used with convicted offenders. It's used in arrestee profiles. So if you're arrested and they take your DNA, they're going to put you into this database. And then they can compare unsolved DNA or DNA from unsolved crimes against that database and potentially find or identify individuals that may have committed that crime. Now, if we take a look at this, CODIS, again, using this technique, the same type of technique that allows you to figure out, who you're closely related to and where your family is from, is allowed to, or is used to match crime scene evidence where they collect DNA with people that had that same pattern of these enzymes, the same bands that appear, and identify them as potential perpetrators or potential people that have committed that crime. And so what they do, they collect the DNA with the evidence, they run it through it in the database. The database, if it has a match, will identify who is the likely person that will have committed that crime. And then they can continue to do an investigation, try to determine if they truly are the person that did that. And so that finishes up kind of our overview of DNA fingerprinting. And a couple different examples using it to determine paternity, taking a look at it as, using it in a forensic science case, or potentially using it to, look at your ancestry, figure out who you're related to, where your family is coming from, all using the same basic DNA technology, but lots of applications associated with it. Now what I want to do for your readings for today's lecture, 2 articles related to the forensic science aspect of it, because they have a better explanation than some of the other applications. So take a look at 30 years of DNA forensics, an article taking a look at how DNA has revolutionized criminal investigations, as well as DNA mixtures, a forensic science explainer that kind of talks about the techniques that we've talked about in today's lecture. As always, if you have any questions, feel free to email me. Thank you and have a great day.